All right, our next speaker is Pascale, who will be discussing the effects of invasive plant removal on an urban plant bee community.
Hi, everyone. Thank you so much for coming out to these presentations and being here. I'm Pascale, and I'll be presenting some of the research that I did for my master's project at the University of Toronto, Scarborough in the Bugs Lab that I defended in February of this year. Let's see if I can get the controls working okay. So this project all started at the Meadoway, which is a 16-kilometer hydro corridor in Scarborough in East Toronto in an urbanized area. And up until very recently, this 16-kilometer stretch was all turf grass with some dandelions and a little bit of garbage thrown in there for good measure. But recently, it has been converted into a native plant meadow. And this project was managed by the Toronto and Region Conservation Authority. And among many beneficial goals of this project, the TRCA was seeking to restore habitat for native bees. Oh, sorry. But like many restoration projects, the Meadoway is up against an enemy, invasive plants. In particular, the non-native creeping thistle, Cirsium arvens, which is a Eurasian Asteraceae that forms these really dense, large monocultures that threaten native plant establishment and thus threaten the success of the Meadoway project. And as such, it's a top management priority at the Meadoway. And the Meadoway team uses a lot of resources to remove this plant through manual and chemical means. And it's a really large-scale removal effort. So this raises questions about potential non-target effects, for example, on the bee community at the Meadoway. And there's reason to think that this might be going on. We know from past surveys that at the Meadoway, creeping thistle is visited by many native and non-native bees. It's considered an attractive generalist flower that has high nectar content that's accessible to lots of different bees. And in natural systems, it's been shown to play a role in shaping the plant bee community structure. And more generally, we know that invasive plants can suppress plant biodiversity and thus suppress arthropod abundance and diversity. So in these cases, the removal going on at the Meadoway might be a positive thing for native bees. But we also know that in cities, especially, invasive plants can be an important food resource for bees because native plants are scarce. And so the communities that live in cities might be kind of uniquely primed to make use of these non-native resources. So in this case, when we remove creeping thistle, we're removing bee food, and that could be negative for the bee community. And lastly, we know that invasive plants can change plant-bee interaction patterns, which, depending on how they do that, could be either positive or beneficial or negative for native bees. To expand on that a little bit more, plant bee systems can be organized in these bipartite networks where the community is mapped out with bees and the plants that they interact with. And these networks can display patterns that inform community stability. So one such pattern is nestedness, where the interactions of the least connected species are subsets of the interactions of the most connected species. And generally, in mutualistic networks, this pattern is considered stabilizing because there's lots of redundant interactions. And attractive generalist flowers like thistle are thought to generally increase nestedness because they interact with bees that have different levels of connectedness in the system. So when we remove thistle, we might be decreasing the nestedness of the network, which could be negative for the bee community. Another pattern that we can look for is modularity. So the tendency of the network to organize in these subsections that interact strongly within themselves and very little between. And generally, this is considered destabilizing in mutualistic networks because the community is easily fractured and there's no opportunity for rescue effects from other modules. Attractive generalist flowers like thistle are thought to decrease modularity because they interact with bees across different modules. So in this case, when we remove thistle, we might be increasing modularity, which could be negative for the bee community. So with all this information, I generated some research questions about what was going on at the Meadoway. First, I wanted to know about the flower community and the food available for bees. So I wanted to know if removing creeping thistle affects the abundance, richness, or diversity of available floral resources at the Meadoway. I wanted to know if removing creeping thistle then affected the abundance, richness, or diversity of the bee community, and if removing creeping thistle alters the nestedness or modularity of the plant bee network. To answer these research questions, we set up an experiment at the Meadoway, where we set up plots in pairs in different subsections where creeping thistle was present. And so at one plot in each pair, we went in and we removed all of the creeping thistle. And then at the other plot, we left it to flower. And then within those plots, in a smaller square in the center, we collected our data. So we collected 3 types of data. First, about the floral community through belt transects, and we collected information about the abundance, richness, and diversity of open flowers during our surveys. And then we collected information about the bee community, its abundance, richness, and diversity, as well as the plant bee network and its parameters of nestedness and modularity through vacuum surveys, where we walked through our research plots and recorded every bee that we saw and what flower it was interacting with. And we conducted this fieldwork over the summer of 2023. So we did a first data collection period before any thistle had opened. So we visited all of our plots over 4 times during this period. And then we did our removal. So at plots where we were removing thistle, we went in and we chopped it all down. At that point, creeping thistle had started flowering. So we did a second data collection period where we visited each of our plots another 4 times. And then we continued removal throughout the season as creeping thistle was regrowing. And we did a final data collection period as most of the creeping thistle had died out. So what did we find? Well, first of all, we found that at its peak, creeping thistle at the plots where we left it made up about 12.25% of open floral units. And then by period 3, when it had mostly died off, it was about 1% of available floral units. But despite this, we found no difference between our removal and non-removal plots in floral abundance, richness, or diversity. So it seems that at the plots where we removed and the plots where we didn't remove, pretty similar availability of bee food. And then we looked at our bee community. So we captured 3,819 bees with our bug vacuums. Most of these were bumblebees, and then non-native Apis mellifera, honeybees, were the second most common bee captured. And then everything else made up about 13%. And we found that before and after creeping thistle flowering, very similar bee community. But during that peak creeping thistle flowering, there were marginally lower bee abundance in the plots where we had removed creeping thistle. So bee communities slightly smaller in removal plots during peak bloom. And then we looked at our plant bee networks. So on the left is an example of what one of those networks look like. For this portion, we only looked at data from the peak bloom and end of season data collection periods because the networks from the early season were just too sparse to have meaningful data. So we found that creeping thistle was visited by 13 out of the 23 genera in our study. We only identified bees to the genus level so far, and we found that no genus exclusively visited creeping thistle. And then we looked at the network structures. And first of all, we found no difference in nestedness between our removal and non-removal plots at any time point. But we did find differences in modularity. So during the start and peak creeping thistle period, that period 2, we found that modularity was significantly lower in the removal plots during creeping thistle bloom, which is not what we were expecting to find. And then later in the season, we saw a non-significant but opposite difference emerge. So what do we make of that? So now we know that at the Meadoway, plots where creeping thistle is removed offer the same abundance, richness, and diversity of bee food. During peak creeping thistle flowering, they have a marginally smaller bee community, and they also have differences in network structure, so they're less modular during peak bloom. So that tells us that bee communities with and without creeping thistle are different, and therefore may be vulnerable in different ways and at different times. And with that, we can generate some action recommendations. First of all, for restoration practitioners to be aware that the bee community is potentially smaller and more fragile during peak creeping thistle bloom in the year of removal. So at that time in particular, it could be important to limit actions that further harm bees. And we can maybe think about replanting with phenologically, morphologically, and nutritionally similar natives. And then for researchers, as we continue to study this question, we're going to be looking for species-specific interactions, so potentially species that interact with creeping thistle. And we're hoping to do this with the data that I've collected as we move through the species-level ID. And then we can also look at more specific nutrient provisioning from creeping thistle to figure out what it's actually providing our bees with. And hopefully with this information, we'll be able to better support urban bee communities at restoration sites going into the future. So that summarizes my talk. And I'd just like to acknowledge some of the folks that made this possible. There was a huge team of people that helped me with this project. In particular, in the McIver lab, my supervisor, Dr. Scott McIver, provided a huge amount of support throughout, as well as everyone else in the lab, in particular, Dr. Nicholas Sukan, who really held my hand through the whole statistic process of this presentation. All the faculty at UTSC who provided feedback throughout the course of my work, and the huge team of undergrads who helped me collect my fieldwork data on some really hot days. It was also the wildfire season and smoky days, and they did a huge amount of really precise work. And finally, the team at the TRCA, Katie, Chris, and Sarah, who also provided support and feedback. It was a huge privilege to conduct my master's research in such a beautiful place, and I'm really grateful to everyone who made it possible. And with that, congratulations.